Okay, good morning, everyone. I am Tiffany Ho. I'm an assistant professor in the Department of Psychiatry and Behavioral Sciences. We are extremely pleased to welcome Dr. Johnna Swartz, who is an assistant professor in human ecology at UC Davis. Prior to joining faculty at UC Davis, Dr. Swartz completed her PhD with Dr. Christopher Monk at the University of Michigan and her postdoctoral training at Duke and UNC with Dr. Ahmad Hariri. Dr. Swartz's primary area of research focuses on how interactions between multiple levels of analysis, including the brain, physiology, behavior, and the environment, predict the development of depression and anxiety disorders during adolescence and emerging adulthood. She is also a member of the International Research Consortium, Identifying Depression Early in Adolescence, which aims to identify risk factors for depression in adolescents across the world. As part of her work on this project, she is collaborating with investigators in Brazil to conduct a longitudinal examination of brain function in adolescents who are at risk for depression or who currently have depression using functional magnetic resonance imaging. So please join me in welcoming Dr. Johnna Swartz. Before we start, I just want to remind everyone that there will be a question and answer session at approximately 9:20. So if you have questions, please submit them into the public chat box. Thank you. Okay, Dr. Swartz, I will hand it over to you.
Okay, so thank you for that introduction. Hopefully you can still hear me okay. And just thank you for the invite to be here today and to talk about a new project I'm really excited about. I have some fairly preliminary results from our study to show you today. So I'm excited to get to talk about this project that we're working on in Brazil, as Tiffany's introduction mentioned. Before I start, I just want to say I have no conflicts of interest to disclose. And I did want to just briefly mention the consortium that I'm a part of, which is called IDEA, or Identifying Depression Early in Adolescence. This was initially funded with a grant from the British Foundation, MQ, which stands for Mental Health and Quality of Life. I won't go into details on the grant or how this collaboration got funded, but if there are questions about how I developed this collaboration with Brazil and other international investigators, I'd be happy to talk about that in more detail in the Q&A, because it's kind of an interesting story about how this got started. But it got started with this grant. And so our consortium, our collaboration, involves a number of international investigators, so many that I couldn't fit them all on one page, but these are some of the kind of key principal investigators and key collaborators. And I just really wanted to highlight Christian Keeling on the left of this slide. So he's a child and adolescent psychiatrist who's at the Federal University of Rio Grande do Sul in Porto Alegre, Brazil. And so he's been critical in this collaboration. So he's the one kind of running the study in Brazil locally, and has made all of this research that I'll talk about today possible, and has really led a lot of this in Brazil. So all of these collaborators have played a huge role in this, and especially want to highlight Christian Keeling, and I'll basically be bragging about a lot of the work he's done kind of in the first half of the talk to set this up. So today I had kind of three points I wanted to cover in the talk. So the three main points are going to be discussing the representation and really the lack of representation of participants from low and middle income countries, or LMIC, in research. Second point is talking about the work of our IDEA consortium or collaboration to develop a risk score calculator to identify adolescents at high risk for depression across the globe. And then the third point is kind of my main role on this collaboration or project, which is I'm going to talk about some results of a study we've done to look at the neural correlates of risk for depression and current depression in a sample of adolescents from Brazil. So kind of starting with the first point, talking about representation of participants in our research. So this figure is from a paper written by Christian Keeling, my collaborator I highlighted a few slides back, where he's kind of taken the lead or one of the leads in the field on raising this issue of lack of representation of low and middle income countries like Brazil, LMIC, in research. So in this slide, he's showing the number of randomized controlled trials, so RCTs, that have been conducted to test treatments for child and adolescent mental health disorders. And he's showing the number of RCTs that have been conducted in high income countries, HIC, like the US. And the color coding is showing you the types of treatments that were tested across these randomized controlled trials. So we have psychosocial treatments, pharmacological treatments, other types of treatments. And these are the number of RCTs conducted between 2001 and 2010. So you can see there's a lot of RCTs being conducted on treating child and adolescent mental health disorders in high-income countries like the US. And in comparison, the bar here is showing you the number of RCTs that have been conducted in low and middle income countries in that same time period. And as you can see, it's a far lower number of treatment studies. And then that's contrasted with the gray bars on the right, which are showing you the kind of relative proportion of the world's population living in these different countries. So this gray bar is showing you the number of people younger than 18 years old, so essentially the number of children and adolescents who live in high income countries like the US and Canada and European countries, as compared to the number of children and adolescents on the right who live in low and middle income countries. So while 90% of the world's children and adolescents live in low and middle income countries, you can see the majority of our research is being conducted in high income countries, and very little is being conducted in low and middle income countries. So that was a study, again, that Christian Keeling wrote. We followed this up pretty recently. So this was actually just published, I think like about a month ago in Neuroimage. And this was led by Christian's grad student, Lucas Patel, and I collaborated on this paper where we kind of wanted to look at a similar question of the representation of low and middle income countries in research. But so on the previous slide, I was showing you treatment studies, randomized controlled trials. In this paper, we looked at research that used neuroimaging, specifically MRI, to examine the neural correlates of adolescent depression. So kind of very closely related to what I do. So we took a similar approach where we did kind of a systematic review, and we looked for any studies that used MRI to look at the neural correlates of adolescent depression. And then we just looked at where those studies were being conducted, and we found very similar findings. So 82% of the neuroimaging studies on adolescent depression were conducted in high-income countries like the U.S. or Australia was a big country where some studies were coming from, and then only 18% were conducted in low and middle-income countries. And then again, that's contrasted with the population of the globe, where the majority of adolescents are living in these low and middle-income countries. So hopefully that convinces you there's this gap in research, both in terms of studying treatments and also in terms of studying kind of risk factors and biological factors associated with adolescent depression and mental health in low and middle income countries, and that there's a need for representation from participants from these countries, given that that's where the majority of adolescents in the world actually reside. And I think this should be of concern even to researchers and practitioners who are in the US, who are only going to research or practice with individuals in the US. Because even though adolescents from the US are pretty well represented in treatment and neuroimaging research, we're still likely missing underrepresented populations. So for example, we know that lower income populations are less likely to participate in research. Sometimes it can be because it's a challenge for them to get to the lab by a certain time or get transportation. We know, for example, some immigrant populations may be less likely to participate in research, and some minority populations are less likely to participate in research. So even in the US, our research tends to include more kind of white, higher income populations. So I think by trying to diversify participants in our research, not just in the US, but across the globe, we can start to ask questions about how much these risk processes and these treatments kind of generalize to these diverse populations. And I think that can benefit people both in the US and in other parts of the globe. So that was my kind of first point of the talk, was talking about the need for more representation of participants from low and middle income countries in both our research on the kind of neurobiological mechanisms of depression and then also treating depression. So that was one of the goals of our IDEA consortium or collaboration, was to increase the representation of participants in research from low and middle income countries. A second major goal was to try to improve our ability to identify adolescents at high risk for depression early on, kind of before a severe disorder develops. So we can potentially improve preventions for depression and also better understand mechanisms and processes of risk. So what is increasing risk or leading to the development of depression? So that was a kind of second goal of our collaboration. So when we're thinking about how we want to define risk for depression and the best way to identify risk for depression, at least in speaking about research first, because that's kind of my world, is I don't do treatment, but just thinking about how we define risk in research. And I'm going to just close my, well, I think it'll be fine. Sorry, just closing my email really quick because I think it's going to just buzz. Okay. And can you still see my screen? That's okay. Research has often defined risk for depression as kind of a binary based on one risk factor at a time. So for example, this is something we've done in some of my prior research looking at neural correlates of risk for depression. So in one of my prior studies, when we looked at this, we defined risk for depression as having a family history of depression. So we looked at a sample of adolescents. Half of them had a family history of depression. So we called this the high-risk group, who we considered as being at high risk for developing depression. And then half of the participants had no family history of depression. So we called them the low-risk group. So this is a pretty standard kind of binary risk approach where you take one risk factor, and then the participants that have the risk factor are high risk, and the participants that don't have the risk factor are low risk. But this approach may lead to a somewhat heterogeneous low risk group, because even if the participants who have no family history of depression, they don't have that risk factor, they might still have other risk factors. So they might be living in poverty, or they might be the victim of bullying at school. And so you might get a somewhat noisy low risk group because you're not capturing some of these other risk factors that might be present for those low risk individuals. So we know depression is really heterogeneous, and we know that it has a number of different risk factors. So there's multiple different types of risk factors that have been identified for depression, and different individuals may experience different risk factors, which may all place them kind of at risk for depression. So research and practice may benefit from the development of a multivariable risk score for assessing depression risk. So moving beyond kind of research that looks at one risk factor at a time, if we kind of combine all the risk factors together into a single risk score, does that kind of buy us anything in terms of predicting risk for depression, and then also kind of categorizing risk for our research on neural mechanisms of risk for depression? And so that was one of the goals of our consortium, was to develop more of a multivariable risk score that incorporates multiple risk factors at once. And Christian Keeling, my collaborator, was really the lead on this aspect of the project, on developing the risk score. And he was really kind of inspired by the Framingham risk score for cardiovascular disease, which takes multiple different risk indicators and essentially kind of computes a risk score that indicates someone's risk for cardiovascular disease. So taking that same approach, but using it to predict depression. So again, Christian and his team were really the leads on this part of the project of developing the risk score. And so the approach they used is to test their risk score initially. They used an existing longitudinal cohort of participants from a city in Brazil, Pelotas. So this was the 1993 Pelotas birth cohort of a longitudinal study of participants followed from birth. They used data collected at age 15 and at age 18 from this longitudinal study. And so they looked at which predictors collected at the age of 15 predicted the development of a diagnosis of major depressive disorder at the age of 18. And so I'll go through, so they ended up developing an 11-item risk score that can essentially be used as a risk calculator, where the more kind of risk items you kind of indicate on your survey or your questionnaire, the higher your risk score. And this was tested with the logistic regression. So the risk factors can also be kind of weighted differently depending on the sample you're working with. Before I talk about the risk score items, though, there are a couple kind of caveats or limitations or factors that went into selecting these, because I wanted to mention these because you might see the risk score items and think like, wow, there's some obvious ones that are potentially missing. So what went into selecting these risk score items in particular? So there were some kind of factors or limitations, again, that went into what was put in this original risk score calculator. So one obvious limitation was availability of the data in that Pelotas birth cohort where this was originally tested. So we had to include only risk factors or variables that were examined in that Pelotas birth cohort in order to test them in that initial study. So it was kind of limited on what data was already collected in that case. Christian and his team used scientific literature reviews and clinical expertise to select based on the variables available, which ones made the most sense to include in this risk calculator. And then a big factor that went into kind of selecting items was his goal was to make this a risk score calculator that could be administered directly to the adolescent themselves, where the treatment professional or the researcher, whoever's administering this, could get all the information they needed directly from the adolescent, and not need collateral information from a parent or a teacher or anyone else. So there are some risk factors, some obvious ones that are missing from this risk calculator, with one of the obvious ones being family history of depression. So as I mentioned a couple of slides back, we know family history of depression is a strong risk factor for developing depression in children of parents who have a history of depression. But they deemed that this might actually be difficult for adolescents to answer accurately, because adolescents might not have good knowledge about their family history of depression. So for example, if they have a parent with a history of depression, their parent might not have shared that information with them. So that might be a risk factor where you potentially might need more collateral information from a parent. And so they didn't include it in this risk score in order to, again, have kind of all items that could be accurately assessed from the adolescent themselves. All right, so heading into what ended up in their final risk score that they tested in the Pelotas cohort. The first risk factor was biological sex, and we know from prior research that girls are at higher risk for developing depression compared to boys. So female sex was the risk factor. Skin color. And so skin color in this item means essentially belonging to a racial or ethnic minority group for the country you live in. So the intention was that this could be potentially applied to different countries and different geographical settings. And obviously, who's considered a racial or ethnic minority will depend on which geographical setting you're in. So having a skin color or a race or ethnicity that's considered a minority kind of status for your location. Drug use. So this could include a range of different drugs that they asked about: tobacco, alcohol, marijuana, etc. School failure. And most of these items were asked with kind of just a single question, like, have you ever been held back a grade or failed a grade at school? Social isolation, involvement in fights, poor relationship with mother, poor relationship with father. And again, this is all just the adolescent's self-report. Poor relationship between parents, childhood maltreatment, and ran away from home. So these were the 11 kind of risk factors that went into this multivariable risk score. All right, so when they tested it, and they've tested it now across several different cohorts, and some of these are still in prep, so I don't have the references for those, but for the ones that have been published, the references are listed here if you'd like to follow up with them. So they tested it obviously in the original Pilotas birth cohort, which is where the risk score was developed. And what I'm showing you is the C statistic. And so this is essentially kind of like a statistical indicator of model fit. So how well the model is predicting depressed versus not depressed status in the participants. Obviously, statistics closer to 1 would indicate better model fit and better prediction. So not surprisingly, the model did the best in the Pilotas cohort where it was originally developed. So the C statistic here was 0.78. And just as a comparison is the C statistic for the Framingham cardiovascular disease risk score is 0.76. So pretty good fit of the model, pretty good prediction of depression risk. And then they went and tested it on a number of different cohorts from located across the globe. So we have one from a different city in Brazil, Sao Paulo, New Zealand. This was the Dunedin, New Zealand study, if you're familiar with that. The UK, Nepal, Nigeria, USA. And so without going into too much detail on this, since I do want to have time to talk about my neuroimaging part on the project, the overall picture is that in all of the cohorts, the risk score predicted depression risk above and beyond chance. So they all performed above and beyond chance, but possibly not surprisingly, as they started testing this in different cohorts, the fit of the model did get worse. So the fit of the model did go down. So it didn't work as well in the other cohorts as it did in the original one. And what they found kind of looking at individual risk items is some of the items seem to be kind of equally predictive across the different cohorts. So for example, female sex was pretty consistently predictive of increased depression risk across the different cohorts, whereas some of the risk factors did not seem to be as predictive of depression in other cohorts. For example, the getting into fights risk factor was predictive of depression risk in this original Pilotas cohort, but was not as predictive of depression risk in some of the other cohorts. All right, so our collaboration has called this risk score that's being developed the IDEA-RISC score, or IDEA-RS. It's a risk score that was developed to estimate risk for developing depression in adolescents. It's obviously still a work in progress, so it's definitely, not finalized, not ready for prime time, still needs to be refined, but that's kind of the ultimate goal. It has been shown to predict risk for depression beyond chance in a range of cohorts across the globe. So there's, some initial support that we could develop this risk score that could be used across different parts of the globe to identify adolescents at high risk for depression. But the research does suggest that the model has poorer fit in some areas for some cohorts. So we'll likely need to calibrate the risk score and adjust some of the risk indicators depending on the cultural and geographic context. So we might need to take out some risk factors for some cohorts and add in some risk factors for other cohorts. And this suggests why we do need to conduct this research across different cohorts and across the globe, because it tells us what are the universal risk factors that are pretty consistently associated with risk across the globe? What are the more kind of cultural or geographically specific risk factors? What needs to be kind of calibrated or changed to work in different settings? So this is obviously kind of a long process here, but this is the kind of current status, and the future goal is to get a risk score that is calibrated to different settings so that it's kind of accurately predicting risk for depression equally well across different settings. And that, again, might include changing some of the risk factors, changing the weighting on these to get this to apply across different settings. And then for the third part of my talk, I wanted to talk about my main role in this project, which is using this risk score to recruit a new sample of participants in Brazil to examine neural correlates associated with risk measured on this risk score. All right, so once Christian and his team had developed this IDEA-RISC score and had tested across the different cohorts, we then used the risk score to recruit a new sample of adolescents in a different city from Pelotas, where it was originally developed. So these adolescents were recruited in Porto Alegre, Brazil, so a different part of Brazil. And I'll kind of skip the details of the school screening process because in the interest of time to get to the findings, but that's kind of an interesting story as well. So if you want to hear more about the school screening and how Christian and his team actually recruited this final sample in Brazil, I'd be happy to talk about that more in the Q&A. So long story short about the screening, they managed to recruit this cohort, which we've called the IDEA-RISC Stratified Cohort, or IDEA-RISCO. And this cohort has 3 groups of adolescents who are stratified based on risk using that risk score calculator that I just discussed a couple of slides back. For our final sample for our neuroimaging study, we had 50 participants in each group. So 50 low risk or LR, 50 high risk or HR, and then 50 MDD or depressed participants. And these were the ones, again, who underwent a neuroimaging or an MRI scan for our study. For eligibility criteria for the low risk group, these adolescents, and I should mention the age range was 14 to 16 to kind of span that 15-year age, which is where the original Pelotas kind of risk factors were measured, was at age 15. So these adolescents were 14 to 16. So in order to be eligible for the low risk group, adolescents had to be below the 20th percentile on that IDEA-RISC score, which I just talked about. So essentially indicating absence of most of those risk factors, so considered to be at very low risk for developing future depression. And then it was confirmed through a structured diagnostic interview with a child and adolescent psychiatrist that they had no current or prior depression, so no current or prior major depressive diagnoses. For the high-risk group, again, this was another 50 adolescents. They had to be above the 90th percentile on the IDEA-RISC score, and also verify through a structured diagnostic interview with a psychiatrist that they had no current or prior depression. So in this group, we have a very high risk group of adolescents who are endorsing a lot of the risk factors for depression, but they are currently and have never previously been depressed. And I can talk about more about that risk group also in the Q&A. And so in our third group, we wanted a comparison of currently depressed adolescents. So that was our MDD group. They had to be above the 90th percentile on the IDEA-RISC score as well. The idea was that so they'd be at a comparable level of risk as our high risk group, so we could kind of more directly compare them. And they had to have a current diagnosis of major depressive disorder confirmed by the child and adolescent psychiatrist for this study. Okay, so once we recruited this risk stratified cohort of adolescents, they all completed a neuroimaging scan. The ultimate numbers ended up being a little bit smaller for the data we could analyze because we always lose a few participants due to excessive motion and quality control criteria. But we had roughly 50 participants in each group. So the participants underwent a pretty standard scanning protocol, and this was at the MRI scanner at the hospital in Porto Alegre, Brazil, that was affiliated with the university where Christian is. So participants underwent a structural MRI scan, 2 tasks, and then a resting state fMRI scan. For the results I'm going to talk about today, I'm going to focus on the results for the tasks because we're still working on analyzing the resting state results. So I don't have any preliminary results for that yet, but hopefully we'll have that soon. So for our 2 tasks, we selected 2 tasks that had been used in prior research on adolescent depression in US or other high-income country samples. to facilitate some comparison with prior research. Obviously, it's not going to be a perfect comparison since our methods were a little different, but we did want to use tasks that had been used somewhat frequently in prior research. So our first task was a gambling task. This task is designed to tap reward-related brain function. So it has conditions that involve reward and punishment. In this case, the reward and punishment is winning or losing money. So that's a great way to reward participants, is to make them think they're winning money. On this task, participants first saw a question mark, and then they were asked to guess whether or not the number behind the question mark was higher or lower than 5. If they guessed correctly, they saw this green up arrow, which meant reward feedback. If they guessed incorrectly, they saw this red down arrow, which meant punishment feedback. So reward meant you were going to win money for that trial. Punishment meant you were going to lose money. And then neutral trials were if the number behind the question mark was 5, you neither won nor lost money. So participants were told this was based on correct guessing, but of course the feedback was fixed. So all participants received the same exact number of feedback in terms of number of reward and punishment trials on a fixed schedule. These were blocked into blocks that were primarily reward blocks, where participants were primarily winning money, and then blocks where participants were primarily losing money. So we looked at the contrast of reward versus punishment, or winning versus losing money. The second task we chose was a face matching task. Again, this was based on a lot of prior research that had linked amygdala activity in particular to altered amygdala activity to emotional faces to depression. So in this task, we had both face matching and shape matching control condition. So this is an example of the shape matching control condition. So participants were shown a target shape on the top here, and they were just asked to select which of the shapes on the bottom row matched the target shape on the top. So obviously in this case, the correct answer would be the left shape. And so it was just a simple perceptual matching task. And then we had similar trials, but that included emotional faces instead of shapes. And so those emotional faces were blocked into angry, fearful, happy, sad, and neutral face blocks. Okay, and then we had the resting state scan, which, as I mentioned, we're working on analyzing, so I don't have that data today. Okay, so here are some of our, again, fairly preliminary results. So this paper is currently under review, and we're working on revisions for this right now, and has been led by my postdoc, Lee-Hyun Yoon. So these, again, are fairly preliminary results that are still undergoing the revision process. And these are not the only results we observed. I'm just picking a few to highlight due to the interest of time. So one of the findings that we observed was during the reward task. And again, our main question we were asking here was, are there differences in neural function between our 3 groups? So we had the low risk group, the high risk group, and then the currently depressed group. So we just did kind of a whole brain group analysis looking for any differences across the brain across these 3 groups. So we found a significant difference in the lateral prefrontal cortex, and this is during, again, that gambling or that reward task where you're winning or losing money. And the contrast here was those reward blocks where you're mostly winning money compared to the punishment blocks where you're mostly losing money. And so there were kind of 2 effects, one in a more dorsal cluster of the lateral prefrontal cortex, one in a more ventral cluster of the lateral prefrontal cortex. The pattern was pretty similar for both of these regions, where the low risk group showed higher activity in these regions during those reward blocks as compared to the high risk and the MDD groups. For the more dorsal part of the lateral prefrontal cortex, we saw kind of more of almost like a dose response effect. So the low risk group showed the highest activity, the high risk group was lower than that, and the MDD group was lower than that. And I'll talk a little bit, I'll talk more about the interpretation of these effects in a couple slides. But it's interesting anytime we see an effect where the high risk group and the MDD group are different from the low risk group, because that could potentially indicate that possibly this is kind of a neurobiological risk factor for depression, given that we see it in both high risk and depressed adolescents, with the major caveat that this is a cross-sectional study. So we obviously can't use this yet to determine whether this predicts risk, but we are following these participants longitudinally, so we will be able to see how these patterns of brain function predict changes in depression symptoms over time. So that was one of our findings. For the FACE task, we actually did not see any significant differences across the three groups in terms of activity. So just kind of basic activation in response to stimuli. We did not see any differences across the groups. And I should mention, we really focused on the angry and fearful faces because that's where prior research in other kind of US and other samples had suggested that we would observe effects. So that's where we focused our analyses. We did not see differences in activity levels. Where we did see differences was with connectivity, so specifically amygdala connectivity. So with connectivity, we're looking at the correlation in activity levels between the amygdala and a different part of the brain. And because this is a task, we're specifically looking at how the connectivity of the amygdala with this region changes while somebody's performing that specific task. So in this case, it's how the connectivity of the amygdala changed when matching the fearful faces as compared to our shape matching control condition. Okay, so we saw these differences for left amygdala connectivity. We also saw somewhat similar differences for right amygdala connectivity. So I'm not showing this here just to make this a little bit simpler of a figure. So the regions where we saw kind of group differences with left amygdala connectivity that were differently connected with the left amygdala were the right thalamus, the right superior temporal gyrus, the left inferior parietal gyrus, the right precentral gyrus, and the left supplementary motor area. And across all of these regions, we saw a pretty similar pattern of results, which was that the low risk group showed increased amygdala connectivity with each of these regions compared to the high risk and the MDD groups, which generally showed lower connectivity with these regions during the fearful face matching. And again, I'll talk about some potential interpretations of these results in a couple slides. And then lastly, we did one additional analysis in addition to looking at the groups kind of as categorical diagnostic groups, MDD, low risk, high risk. What we did was we combined everybody together into one analysis, and we just looked at correlations with depression symptoms as a continuous measure. So to look at if there were kind of patterns of brain function associated with depression symptom severity. So here in this analysis, we did get one significant effect, and it was again with amygdala connectivity during the fearful face matching in our face task. And we got the same or very similar effects for the left and right amygdala connectivity. In both cases, we found that higher levels of self-reported depressive symptoms, so higher depressive symptoms, were correlated with lower amygdala medial prefrontal cortex connectivity. So more severe depressive symptoms correlated with decreased amygdala MPFC connectivity in slightly different clusters for left and right. So that's for the left, and then that's for the right. Okay, so summary and interpretation of these findings. So we saw a couple differences between the groups when we were looking at these as more kind of categorical group differences. One was in the reward task, where we found decreased activity in the lateral prefrontal cortex in both the HR, high risk, and MDD groups compared to the LR or the low risk group. So the lateral prefrontal cortex is involved in cognitive control. It's also involved in integrating information about reward and also integrating and using this to inform value-based goal pursuit. So kind of top-down driven goal pursuit that's informed by feedback about reward and punishment. So speculatively, since we weren't expecting this finding, This potentially could contribute to decreased motivation and decreased value-based goal pursuit, which we know are characteristic of depression, that decreased motivation and kind of difficulty getting out of bed and pursuing goals is kind of a symptom or characteristic of depression. So that's possibly contributing to that lack of motivation. For the FACE task, we found that decreased amygdala connectivity, so not activity this time, but connectivity, with a range of parietal and temporal regions was observed in the high risk and the MDD groups relative to the low risk group. So these regions in the parietal and temporal cortex play a kind of a range of different roles and are involved in a range of different functions. One of the functions that some of these regions are involved in is mentalizing or theory of mind. So this could involve kind of any mentalizing processes that involve being able to understand the emotions or mental states of others. So perspective taking, being able to read others' emotional cues, being able to understand somebody else's emotional states. So one interpretation of this decreased connectivity could suggest some reduced mentalizing abilities, possibly difficulties with taking others' perspectives or being able to understand others' mental states or read others' emotional cues. So that's one potential interpretation of this. These regions are also involved in attention control and emotion regulation. So decreased connectivity with these regions could also potentially suggest decreased attention or emotion regulation, which could contribute to symptoms of depression associated with heightened negative emotions. And then when we looked at correlations with depression symptom severity, we found that higher depression symptom severity was correlated with decreased amygdala MPFC connectivity. We know that these connections between the amygdala and the medial prefrontal cortex are really important for emotion regulation and for regulation of the amygdala. So it's thought that the medial prefrontal cortex can send signals to the amygdala to regulate its activity, which allows flexible regulation of amygdala activity, based on someone's current circumstances. So decreased connectivity between the amygdala and MPFC could suggest decreased emotion regulation or decreased kind of regulation of the amygdala, which could contribute to, again, the heightened negative emotions that are symptoms of depression, like heightened sadness, guilt, irritability, negative affect due to difficulty regulating the amygdala. So those are, again, some potential speculative interpretations of our findings. I also wanted to spend a few minutes talking about which of these findings are consistent with the prior research conducted in US samples and other high-income countries, and which of these results are not consistent with that research. So in thinking about what's consistent with research in US samples, I think the finding that's most consistent and that popped out to me when my postdoc kind of and I were discussing these results was this correlation with depression symptoms and amygdala MPFC connectivity. So this finding has been observed in prior research in the US and other high income samples, this connection between depression and altered amygdala MPFC connectivity. And again, this is often interpreted as suggesting decreased emotion regulation and difficulty with regulating amygdala activity. So very speculatively, this could possibly indicate, a neurobiological risk process that does generalize to different populations and may be somewhat more universal. With obviously the major caveat that we did not have a direct comparison group of US or other high-income country participants in this study. So we weren't able to directly compare with a group of participants in the US or other countries. And so we can just kind of loosely compare with prior research, but not directly compare since we didn't have a comparison group. Okay, what is not consistent with research in US samples? A lot of these findings are not consistent with research in US samples. One is our finding of decreased lateral prefrontal cortex activity in the high risk and MDD groups. This was not a finding that we expected to observe in this study. It was not one of our hypotheses or predictions because it hadn't really, this finding hadn't been observed in prior research in depressed groups in other countries. So that was one difference that we observed that hadn't been observed in prior research. And kind of what struck me the most was some of the lack of significant differences that we got in certain regions, especially the ventral striatum for the reward task and the amygdala for the faces task. So we hypothesized, and I actually really strongly predicted based on prior research in US samples that we would get, big effects for these two regions in this study. So a lot of prior research in US and other high-income country samples had suggested that depression is kind of closely tied to blunted ventral striatum activity. And the idea there is that the ventral striatum is important for anticipating and experiencing reward and pleasure. And so if you have blunted activity in that region, that could explain the anhedonia that is characteristic of depression. So we expected to observe that in our gambling task in the study, and we did not. And then for the amygdala, prior research in other samples had suggested that depression is associated with increased amygdala activity to negative emotional faces, so angry and fearful faces. And that's thought to, again, contribute to the heightened negative affect and potentially some of the biased interpersonal processing that occurs in depression. But again, we did not see differences in amygdala activity in this sample. We did see differences in amygdala connectivity. So we didn't see these differences or these correlations with depression symptoms that I hypothesized or predicted we would observe. But there's many different potential explanations for why we got these different effects. And some of these are just come down to the differences in methods in our research as compared to prior research. So there are some obvious big differences in our did not mean to move forward. So there are some obvious big differences in our methods. One is that we used this risk score calculator to define risk for our study, and that's not typically how risk has been defined in prior research. As I mentioned, the most common method of defining risk for research is using family history of depression as the sole risk factor. So we defined risk in a very different way. We also required participants in our MDD group to have very high levels of risk on our risk score, which is somewhat unusual for studies. So usually when studies have a case group of major depressive disorder, they don't have a requirement that they're exposed to a certain level of risk to be included in that group. As long as you have a diagnosis of major depressive disorder, that's enough. So our MDD group might have been a little bit different than MDD groups in prior research. And then there are obvious differences, like we had a different fMRI scanner, we had different acquisition parameters, slightly different kind of task protocol and procedure than prior research. So I think the next obvious step is to, again, have a direct cross-country comparison where we have a group of adolescents either recruited in the US or Europe or some other country using the exact same kind of recruitment procedure and eligibility criteria and the exact same scanning protocol. So we can really more directly compare and really kind of at least remove these research differences as potential explanations for these differences in findings. And then if it's not differences in the research protocol that are leading to these differences in results, then it could possibly be true cultural or geographic differences where we're really seeing that there are different neurobiological mechanisms and processes of risk in this sample as compared to the US sample. And that could be due to a variety of factors that we could then explore. So there could be genetic differences, there could be cultural differences, there could be differences in how depression is experienced, or how participants think about these depressive symptoms, or which ones are the most key symptoms in this culture or region. And I can talk about that more in the Q&A if that's of interest. So there could be, again, kind of true geographic or cultural differences in some of these neurobiological mechanisms that we could get at more with, again, a more kind of direct cross-country comparison. All right, so future directions. As I mentioned, we're currently planning to follow these participants longitudinally to conduct a follow-up wave of neuroimaging and assess future diagnoses of major depressive disorder in these participants. And we plan to examine whether the patterns of brain function we've identified here or any changes in brain function that we see over time are associated with the development of major depressive disorder later in adolescence. And this longitudinal follow-up has actually been funded already by NR21 from NIH, but unfortunately, there's actually been a massive surge in COVID-19 in Brazil right now. And so that has actually prevented us from being able to start this research, but we're hoping to eventually start it sometime possibly within the next year or so. And then the long-term, as I mentioned, future direction would be doing a more kind of direct cross-country comparison. So takeaways and conclusion. So in this talk, I described the IDEA-RS or IDEA-RISC score, which is kind of still in development and being tested. So it's still kind of in the development process. Currently, it includes 11 indicators that can be reported by an adolescent directly and has been shown to predict depression risk above chance across different cohorts around the world. Our neuroimaging research in a sample of Brazilian adolescents at risk for depression based on this IDEA-RISC score suggests some patterns of neural activity that are consistent with depression research in the US, specifically that finding of altered amygdala prefrontal cortex connectivity, and others that are not consistent with US samples. So hopefully I think this suggests or indicates that there's a clear need for more representation in research of diverse populations, more cross-country comparisons, so we can really get a sense of what are the kind of universal risk factors and possibly universal treatments that tend to be effective across diverse populations of individuals, and what are more kind of culture or geographic specific risk processes so that we can kind of better understand risk and then also better treat depression across a diverse range of participants within the US and then across the globe as well. So I just wanted to acknowledge all of the funding and the grants that went into this project and my collaborators, again, who I mentioned at the beginning. And that is all I have for you. So thank you.
What a fabulous talk. Thank you so much, Johnna. So we have some time now to go over questions. So I'll read through the questions that were submitted into the public chat window. The first question is from Tony Young. He's very interested in receiving more information about the IDEA Consortium. I think he had to leave to see patients, so perhaps you can connect with him later via email and give him more information about how one may possibly join, which could be good in terms of being able to take the tasks that you are running in your current study and having a comparison group of more high-income adolescents and seeing if you would find similar or different results.
Yeah, I'll also mention Christian's very interested in testing this risk score again across different cohorts just to see, what are the risk factors that are and are not predictive. So if anybody has a cohort available, roughly 15 to 18-year-old kind of longitudinal that you'd be interested in testing this risk score on, I'd be happy to connect you because he's always interested in again, kind of testing the generalizability of this.
Yeah, that's fantastic. Some questions came up when you were discussing the IDEA-RISC score and, its effectiveness in predicting depression across multiple cohorts in different countries. So the first question is this variance in different parts of the world also true of the Framingham cardiac risk model?
That is a great question. So I actually don't know the answer to that. I'm not sure. I know they have different versions for men and women. So I think there are some differences by sex. I'm not sure if there are differences across different geographic regions, though. Yeah. Okay,
and then the next question, the question I had actually was, how does that C statistic that you presented that's indicating model fit for predicting depression from that composite risk score, how is that related to sensitivity or specificity of prediction? So if the C statistic was low for a particular country or cohort, is that due to not capturing those who are truly depressed, or was it vice versa?
Yeah, so I think you don't get at the answer of sensitivity or specificity with the C statistic. It's kind of just this broad standardized metric that tells you how well the model fit or how essentially kind of if you took, a participant who was indicated as being at high risk, how well does that predict the risk? But I don't think just from that statistic alone, it gives you sensitivity or specificity. So I will say that what they did find was that it does not seem to be entirely specific to depression risk. So one of the things they did in testing this was look at how it predicted other psychiatric problems. So I think they looked at ADHD and OCD and possibly some other disorders as well. And it does predict these other psychiatric problems above and beyond chance as well. Not as well as depression itself, but it does seem to be not entirely specific to depression. And so one of the questions that we have, or the potential compounds or factors in interpreting our neuroimaging results, is we don't know specifically what this high risk group is at risk for. So they are at risk for depression, but they're possibly also at risk for ADHD or OCD or some of the other psychiatric disorders that were also predicted by this risk score. And so it's not entirely specific to depression, and that could be one of the potential limitations. Yeah.
That's an excellent point. I mean, many of the environmental factors that were highlighted in that composite risk score are indeed related to both internalizing and externalizing psychopathology. And they may be risk factors for just general psychopathology later in life.
Exactly. Yeah, I think it's really challenging to find risk factors that are specific to depression versus some of these other. And I think that's the big $1 million question is, could we get more specific to these specific disorders, or is it just going to tell us high risk for kind of a range of internalizing and externalizing outcomes?
Mm-hmm. I think related to that, so Descartes Lee is asking, how is depression defined? And in one of your studies, you did state that it was defined or diagnosed with a child and adolescent psychiatrist. But I'm wondering, for that larger study, when they're testing this composite risk score in different countries, was depression defined differently? And is that part of the reason why the model may not do as well compared to other psychopathologies or syndromes where there appears to be more of a consistent presentation across the world?
Right. That's a great question. Yeah. So because I did not lead this work, I'm not 100% certain. I believe all the cohorts they tested, a requirement was that they did have to have a structured diagnostic interview for depression. So for example, like in the Dunedin cohort, I know they do structured diagnostic interviews. So I know they based it off of that. So I think most of the cohorts they examined did have a structured interview kind of as a requirement for testing it in that cohort. But it's a great point about differences in how depression is defined. And they've also been doing a lot of interesting work kind of in the same idea risk score sample, the risk stratified cohort, in terms of looking at, as you mentioned, the different presentations of depression. So I think one of the difficulties is that depression is so heterogeneous. So two people could be depressed, but really look completely different in terms of behavioral profiles and their symptoms. And so what's interesting about the work, so they did both qualitative work in that stratified cohort to just ask the depressed adolescents, like, what are your, key issues? Like, how are you kind of thinking about this? And they also did network analyses with the symptoms. So looking at what are kind of the hub symptoms or the central symptoms that seem to be kind of the biggest kind of connectors of all the other symptoms, and which symptoms are more peripheral and less central. And interesting, what popped out of that, at least in this Brazilian sample, was that loneliness really popped out of the qualitative interviews. So that was kind of one of the key kind of problems mentioned by the adolescents that was difficult for them. And then the same symptom of loneliness kind of popped out in that network analysis. as being kind of a hub of symptoms and fairly central and fairly common in terms of in depressed adolescents, what was a very commonly reported symptom. And so that's interesting because maybe that is not how, other countries or cultures are experiencing it, or maybe that's not a key symptom. I think that's an empirical question because I don't know, how much this research has been done or compared. But it does ask that question of is depression kind of different or experienced differently, or are the key symptoms different across these different cohorts? And I think that's definitely something they're planning to look into more.
Well, that's really great. I mean, it's so great to hear that kind of work. I mean, it's much needed in the field.
Yeah, I think it's great to also just, look at like what are the key symptoms that are troubling the participants in terms of thinking about treatments and what to focus on.
Yeah, absolutely. I mean, there may be subtypes or subgroups, and those subgroups, based on their presentation or constellation of symptoms, may have different neural underpinnings. So that could be something to follow up with. All right, and then the last two questions, Peter Stewart asks, the risk factor scores seem to measure environmental exposures. Could one argue that depression, whether a risk factor or neurobiologically based, is functioning as an injury signal to those environmental exposures? The differences in findings might also suggest cultural differences in how those risk factors are experienced.
Yeah, I think that's a great point. I think sometimes we have to think about, are some of these reactions to these risk factors kind of adaptive given the risk context that someone is in? So it's often mentioned, like when we see patterns of heightened amygdala activity, often in response to some of these stressors, even though we didn't see this here, that's possibly adaptive, because if you're in a dangerous setting, then it's adaptive to have this overactive threat response to keep you safe. So is this either kind of are some of these an adaptation to the context they're in, or kind of an injury signal, or kind of a signal that to socially isolate, maybe because relationships have not been very protective for you? And I think that's a great question and an interesting way to think about it. And I think a way we should think about it more is if we understand kind of how these might be adaptive or functioning, then that gives us maybe different avenues towards intervention. I do think these are also potentially cultural differences in how these are experienced. And that's probably why we didn't see some risk factors applying in other cohorts. Like we didn't see the involvement in fights predicting risk for depression and other samples, and those could be kind of cultural differences in terms of how common that is, or what that means, or differences in social kind of relationships. So I think those are, both great ways to think about it.
Oh, great response. All right, last question by Julio. The risk score items seem to aggregate phenomena from all three biopsychosocial realms. So for example, trauma history, biological sex, skin color. The latter two items are intrinsically biopsychosocial already. Mm-hmm. Presumably, all of these predisposing factors would funnel down to identifiable neural correlates if the research proposes to find them. Any thoughts as to how risks from such different categories, bio, psycho, and social, could lead to such a neural correlate?
Yeah, that's a great question. And I think it brings the point of how do we examine this? So we kind of did a first pass of examining risk in the aggregate. And I think there's arguments to be made for that, but there's also arguments to be made for examining these as separate risk factors, with the idea that separate risk factors might have more distinct effects on neural function, which might be, for example, why we didn't see some of the effects we predicted, is because we just used this aggregate, fairly non-specific measure that involved kind of a range of different, as you mentioned, kind of biopsychosocial and different types of environmental risk factors. And so I think that would be the next step, is looking at maybe some of these individual risk factors instead and seeing, are we getting more kind of predictive utility in terms of depression risk, or maybe more specificity by focusing on individual risk factors? Are we getting more from the aggregate? And are we able to determine any specific links between these risk factors and brain function? So there are some current theories out there, like the McLaughlin theory is a common one, that there are going to be different neural correlates of more threat-based risk factors like violence versus more deprivation risk factors like poverty. Some other research has not held that up and has suggested that kind of any form of risk has somewhat similar effects on brain function. And so I think that's a huge question for our field is, are we actually going to be able to identify these specific patterns of brain function linked to specific risk factors? Or is it going to be more of an aggregate where just the more risk factors you have, the stronger the effects on brain function? So I'm definitely interested in examining that more. And I think that's a great question for research.
Yeah, those are such excellent points, Joanna. I mean, in Kate McLaughlin and Margaret Sheridan's theoretical model of deprivation versus threat, I mean, these things in reality tend to co-occur. And so I think that's also an important thing to consider when examining this work.
Right, exactly. Does it make sense to separate these out? When that's the other thing they found in the network analysis was that these risk factors, again, did tend to co-occur. So for the participants that were experiencing a few of them, they tended to experience kind of more of them. So yeah, it's very hard to, I think, look at them separately sometimes because they
like to purely isolate them. I mean, I think this network approach that you've taken on is such a great way, to handle these types of data.
Yeah, I think they're working on publishing that paper, so that should be out soon if you want to put a little search alert for Christian Keeling. So,
yeah. All right, well, we're over the time, but this was such an enlightening discussion. Thank you so much again for giving such a wonderful, fabulous, and pertinent talk.
Yeah, thank you again for inviting me. I was excited to present this. And then if anybody does want more information about the consortium or anything, I'm happy to just feel free to send me an email. I'm happy to discuss.